Professor, I think you're on mute right now.
So far?
Okay, now we can hear you.
Oh, so far you didn't hear?
No, not until now.
Oh my God. I'm so sorry. I'm talking about myself then.
Okay.
Okay. Yeah. Now you hear, right? Thank you. Thank you.
Yes.
Yeah. Let me. Okay, great. Thanks. So today we are going to, discuss again with the fluorescence microscopy principles and advantages. So what is the advantage of the fluorescence microscopy? We discussed it before, the light microscopy and also fluorescence microscopy. So the fluorescence microscopy is help us to understand further not only the structure but also the cellular targets, what is going on with them and how that affected due to there are several reasons. So we are going to, understand further, apart from the structural analysis, the fluorescence microscopy help us to understand further how, the targets is interact with each other and how the abnormalities and how, other cellular events and how the protein-protein interactions, how cellular trafficking, these are all, the basic, understanding we can, if you use the fluorescence microscopy to get all these details. So one is the advantage of fluorescence microscopy is it's very, very sensitive because, which detects low abundance molecules with strong fluorescence signals. So, like sometimes the proteins are expressed enough means it's easy to detect. But some reasons, due to treatment, due to other abnormalities, due to the mutations, or due to radiations, there are so many reasons the proteins or any target will be low. In that case, if you use the fluorescence technique, the fluorescence microscopy, which the signals we can easily detect using this, fluorescence microscopy. Also the specificity, because here we use the target, directly using the antibody so that you can, target easily whatever protein or other, cell component easily you can identify that, using the fluorescence microscopy. Also the multiplexing. So multiple fluorophores allow visualization of different targets simultaneously. So this is another very important, targets. Usually, like if really interested, we can detect only one target. Then you can use one fluorophore. Then directly you can identify to see the expressions. But if you really wanted to study about the protein-protein interaction or one particular protein that how affect other cellular targets. So if you wanted to go one by one, then you have to take a sections, transfer onto the slides, and add the primary antibody and secondary antibody. Then you can take a look under the fluorescence microscopy. You can see the protein is expressed or not. Then another slides you can take it and process again with the another antibody and then another target, like 3 or 4. That means you are duplicating or triplicating, you repeating the same to detect in different things. But whereas here, one slide, when you transfer your tissues, after the sections, you transfer onto the so that easily you can do all in one slides, and you can detect simultaneously all the your targets. So more than one target you can easily identify using the multiplexing, by using the fluorescence microscopy. Another thing is a live cell imaging, which enables a real-time study of a dynamic process in living cells. So I really wanted to see how this cellular events, how the protein-protein interaction, how the protein trafficking to go into different, target, and how it is interact, and how subsequently it will affect the targets. So these kind of studies, you can use the fluorescence microscopy. You can do these techniques. High spatial resolution can visualize a subcellular structure as well using this technique. Also, the minimal sample damage, less phototoxicity when compared to electron microscopy. So we are going to discuss later this, but when you use the fluorescence microscopy, due to this fluorescence technique, the phototoxicity is very less. Also, you can take an image, after you process. It's very easy and very convenient and fast photograph. Also, quantitative analysis. So this technique of fluorescence microscopy, not only you can see the expression of your target, but also you can measure the intensity level, the fluorescence intensity level. So based on the intensity, you can determine how much that target is expressed. So that is another advantage of this fluorescence microscopy. So this kind of advantages of the fluorescence, you can get more details about your target. Also, you can do, in situ hybridization. That's a fish technique that also gives you more, data for, genetical abnormalities and other aspects. So this kind of study, the fluorescence microscopy, when compared to, bright field microscopy, you get more, informations on the cellular events when compared to the bright field microscopy. So last class, we discussed about these two different approaches in the fluorescence microscopy. So one is like, the direct target. That is, for example, here that is, you can imagine that the target is the antigen is a lactofferrin protein. So, for example, in gingival tissue, the lactofferrin level, how much it is expressed in the periodontal disease when compared to healthy gingival tissue. So what we can do is we collect the gingival tissue, and then you can process, and you can incubate that with the primary antibody, that is anti-lactofferrin antibody, for a few hours, and then you can wash it, and then you can use the secondary antibody that is conjugate with the fluorophore. So now the primary antibody directly binds to the lactofferrin, because this primary antibody is anti-lactofferrin antibody that is raised against the protein, so that it will go and bind onto this. So now we wanted to detect the, these interactions. So now we can use the secondary antibody that is, collected from the same where the species we make, this primary antibody. Then you can conjugate that with the fluorophore. So directly this fluorophore, this secondary antibody binds to the primary antibody. Then the interaction will be go and see it on the fluorescence microscopy. You can see whether these protein are how much it expressed. So another approach is we can have things same here also. It is lactofferrin. So here this primary antibody that is, directly our target is the same, the primary antibody that is anti-lactofferrin antibody that is go and bind onto lactofferrin. So now we wanted to, study about the interaction, how much this protein is expressed. Instead of here directly, we can go to the secondary antibody, which conjugate with the fluorophore. We can conjugate this secondary antibody with biotin. Okay, so next is the biotin. We wanted to detect this interaction, and we can use the streptavidin. This is a bacterial protein. It is avidly binds to the biotin. So now the streptavidin we can conjugate with the fluorophore. So now the interaction is the streptavidin will go and binds to biotin, and then the biotin and the secondary antibody, this interaction will goes to binds to the primary antibody. Then overall, we are going to see under the fluorescence microscopy, you can detect this lactoferrin expression level. So now these two is the same goal you are going to use to study the expression of the lactoferrin. But we are approaching in two different ways, whether that one is a direct target, and whereas here we use the biotin and streptavidin extra things. So what is the advantage of the biotin and streptavidin, and also over this target? So before we talk about that, the protocol is very simple. As the direct protocol already we discussed, that is, the sections, fixation, dehydration, infiltration, embedding, section is the same. If you fix your sample with the paraformaldehyde. For fluorescence microscopy, the ideal is, cryo fixation. If you fix your samples with liquid nitrogen, that is the preservative. Then when you take a sections using the cryo microtome and transfer that onto the slide. So now you can use that sections with the process for direct target, like you can add the primary antibody, secondary antibody, conjugate with the fluorophore is one slide. Then the another slide with the sections you can process is same thing here. So the sample preparation is for both is like we discussed, we have 4% of paraformaldehyde, then permeabilization that will help you to infiltrate this. So using the detergent, and then blocking is reduce non-specific bindings. Incubate samples of the blocking buffer already we discussed is bovine serum albumin. So then now next is incubation with the biotin-related primary antibody. So here you have already we discussed about whether you can use the primary antibody directly to conjugate with the fluorophore. That means you no need for the secondary antibody. Similarly, if your antibody does not binds to conjugate with if you conjugate with fluorophore directly, then you can detect immediately. You no need to secondary antibodies. So here, so here we didn't conjugate the primary antibody with directly biotin or streptavidin. Instead of that, we use the secondary antibody. So here, another approach is directly you can primary antibody, you can biotin related. So two approaches. It's for convenient, we already discussed, and you can, the record you must have, what is the advantage of that. So now here, instead of this primary antibody, the secondary antibody, the biotin and streptavidin is directly conjugated to the primary antibody. And then you can incubate this for 1 hour, minimum is 1 hour. It should be go for 1 hour, 2 hours or 3 hours, or you can keep it in 4 degree for overnight as well. So then you can wash, 3 times with the PBS. So now your target is that, incubation, the fluorophore conjugated the streptavidin, that is, fluorophore conjugate. The streptavidin you can conjugate with the fluorophore. Streptavidin, the Alexa, so that is one thing. So that again, this is 30 minutes to 1 hour. You can incubate these sections with streptavidin conjugates. And then, if you really wanted to study nuclear staining, then you can incubate that again with the DAPI for 5 minutes, and then you can wash several times with the PBS, and then you can wash out and air dry, and see under the fluorescence microscopy. Now you can see whether the lactoferrin is how much it expressed in the cells. So here, similarly, the same protocol we discussed previously, instead of the incubated bioattenuated, the antibody directly conjugate the fluorophores. Okay, so now you have two different approaches, and what is the advantage of bioattenuated, biotin streptavidin, conjugates when compared to the direct fluorophore labeling? So the advantage is strength, the signaling, because due to multiple fluorophores which binds per target, if you use the biotin. So the streptavidin will go and bind into biotin with the multiple sites, so that the signal, the expression you can easily see, even though the target is low concentration. Whereas the direct, when compared to this biotin streptavidin, this is moderate to low. So the sensitivity is very high, even amplifies the signals, whereas here it's a lower. Also the affinity, the biotin streptavidin system, which binds, extremely strong, whereas when compared to the fluorophore directly labeling. Also the photo bleaching resistance, which is also higher fluorophores which protected within the complex, so that due to this continuously you are using the fluorescence lamp, that will bleach your the expression of your the fluorescence, the dye. So, whereas in the direct labeling, fluorophore is more prone to the photo bleaching, because here the biotin and streptavidin complex that is within the target, it can protect. Also reduce the background noise in biotin and streptavidin system if you use, because the specific biotin streptavidin interaction that will be minimize the background when compared to direct fluorophore things, because it's a non-specific binding may occur with this system. So these are all the two different approaches we are working. The biotin and streptavidin system is better, but it's an extra step, and things. Not necessarily you can always use, even though it's a better, you can go direct fluorescence fluorophore labeling is quick way you can use for routine. Also the specificity, advantage of this is high when compared to the direct fluorophore labeling. Multiplexing capacity, in the biotin streptavidin system is very high, because the multiple fluorophores can be used within different biotin-related target. That's advantage of this. Whereas here it's limited, because the direct labeling requires a separate antibodies for each fluorophore. So that is the main one. Also the flexibility, here the same biotin-related antibody can be detected with a different fluorophore streptavidin conjugates. Whereas here it is lower because the fluorophore is fixed to that particular antibody. Also, if you really wanted to study more details and high resolution fluorescence microscopy, the biotin is excellent when compared to the direct fluorescence. Also, there requires how easy to use. Which one is the best to use is the direct fluorophore labeling. Whereas here, it's require additional steps. You can working on biotinylation and streptavidin labeling is extra additional steps. So mainly this background signal from endogenous biotin in tissues that is possible in this approaches, whereas direct fluorescence labeling, that's a lower specificity, weaker signal, and photo bleaching is possible in the direct method. So both are advantage and some disadvantages, but overall the goal is what do you want to do study. So the regular routine is the direct fluorescence labeling is working enough to study. But if you really wanted to study the low molecules and expressions level and the weak signaling, then you can use the biotin streptavidin signaling and also multiplexing and other aspects. So now, understand the basics and now the techniques between fluorescence microscopy and light microscopy. So the purpose of the light microscopy is to, gain more knowledge about the structure of your cells, the tissues. So whereas here the fluorescence is not only you can see the structures, but also you can, measure the target. So the principle for the light microscopy mainly which use visible light is as a light source, whereas the fluorescence microscopy uses fluorescence dyes and proteins. The resolution, always, the light microscopy has a 200 to 400 nanometer, while fluorescence microscopy can achieve higher resolution is approximately 200 nanometer, or better with the high resolution techniques. The contrast mechanisms is the light microscopy, which relies on natural contrast or hematosylin eosin staining, whereas the fluorescence microscopy highlights specific molecules, that is using fluorescence target. So the live cell imaging both can be used, light microscopy and also fluorescence microscopy. For you can use both, but the fluorescence microscopy provides a real-time molecular tracking. That is, when you inject the dye, you can easily monitor how this dye tracking into different cellular targets, and you can measure the, your target based on which one you are interested. The multiplexing fluorescence microscopy allows multiple fluorophores for different targets, while light microscopy, which does not have that. So the application is light microscopy is used for general biological observations, the structural analysis, while fluorescence microscopy is ideal for molecular imaging, immunofluorescence, and advanced cellular events you can detect with this. So resolution, as we see, it's a 200 nanometer in fluorescence microscopy. Light microscopy moderate is a 200 to 400 nanometer. So we'll discuss more details later with the electron microscopy. Live cell imaging both can, yes. The specificity high, fluorescence microscopy, speed also, fast, multiplexing, fluorescence, yes, but not in light microscopy. preparation is both are techniques are same. So this is a basic, the fluorescence microscopy's advantage and the principles. And finally, we can conclude that the fluorescence microscopy is an essential tool in biological and medical research, which provides high-resolution imaging, real-time observation, and quantitative analysis, that making it indispensable for modern scientific studies. So with this, we completed the fluorescence microscopy and light microscopy. So the next is now laser capture micro dissection and its application. So far we discussed the fluorescence microscopy, light microscopy. You can see structure, and also you can see the expression of your target, and this is. So overall, we thought that the microscopy means we are going to see the structure. So does ever we thought that we can isolate cells using microscopy? Anyone guess? So that is the laser capture micro dissection technique. The isolation and molecular analysis of highly purified cell population from complex heterogeneous tissue has been a challenge for many years. So when you collect a biopsy samples, so now the samples is mixed with both healthy versus infected cells, or healthy with the cancer cells or tumor cells, to separate them, for analysis. What is going on with the cancer cells? What is going on with the healthy cells? What is going on with the infected cells? If you wanted to study, we wanted to separate them manually. So now when you do such kind of separations, it's very difficult to separate them pure cells, healthy versus cancer or tumor cells or infected cells, because of contamination. So until 1996, which is a challenge. So first, the LCM, that is a laser capture micro dissection, was developed in 1996 at the National Institute for Health. The LCM is a non-molecular, minimally disruptive method to obtain cytologically and/or phenotypically defined cells or group of cells from heterogeneous tissues. So this technique, you can able to isolate a single cell from heterogeneous mass of or complex cells. So that is the technique's advantage of this technique. This is the principles. The goal is to isolate a single cells from a heterogeneous mass of tissues. So generally, we can use a flow cytometry to identification of different cell types. Also, the manual dissection method, as I discussed now, it's very laborious, difficult to work with the separations. The contamination is possible, and also based on the experience, who handled the tissues to separate. That also it require things, and prone to contaminations. So these are all manually when we do handling the tissues to separate them. So After this technique, we can able to isolate a single cell from heterogeneous mass of cells. We can avoid all these problems. So the application is we can able to do molecular classification of cancer cells based on the gene expressions. Also, we can able to understand the expression profile rather than the structure or morphology. Also, we can study on novel prognostic indicators using this kind of technique. Also, we can predict, the in vitro based response to therapy. Also, if you really study, individualized vaccines or individualized medicine, this technique will help to understand further precisely. So based on these 3 steps to laser capture microdissection is the successful application of LCM is, however, which dependent on 3 critical factors. One is the tissue morphology, capture success, and maintenance of molecular integrity. So to achieve this, tissue morphology means when you collect the tissues, as you remember that the preservation, you have to preserve as quick as possible so that when you isolate this, dissect the samples, you can preserve the structure and the antigenicity as quick as possible. That is the main goal. So otherwise, it will, go for degradation of, your targets, or the structural integrity will be, go wrong because, due to our, improper handling, contamination, dehydration. There are so many things that happen if you are not properly processed the cells. So the main critical factor is to achieve a single cells is based on the preservation, then how you are going to collect the cells or isolate the cells. And also during all this process, how that will maintain the molecular integrity. So all 3 critical factors depends on how you are preparing your sample, how you are dissecting your cells, and how you are going to analyze the sample. So this is very, very important for laser capture microdissection technique. So here, standard, the microscope is, the laser capture microdissection is like how we integrated the fluorescence, components to the light microscopy. Similarly, either fluorescence microscopy or bright field microscopy. Some of the components, if you attach it to this, then we call it as a laser capture microdissection microscopy. That is, standard inverted microscope and the stage. Then the extra is control unit, and then it adds a low power infrared laser. Then the specimen transfer device and caps with the thermoplastic film. These are all some components that is attached to the fluorescence microscopy or light microscopy. We can call it as laser capture dissection. So this is typical laser capture microdissection microscopy. Everything is the same as a fluorescence microscopy, except this one is the light source, xenon or mercury, that is a light source, because it shows because it's a protect the laser. So otherwise everything is the same. Things, the devices, when you look at this area, that is to collect the tissue sample. So here, the monitor we attach to this is, you can monitor before and after you can dissect the cells. So there are three different type of laser capture micro dissection microscope is available. One is infrared based system. The other was UV or combined. That's the system is commercially available. So this technique is visualization of the cells of interest through your microscope. So when you look at this under the light microscopy or under the fluorescence microscopy, you can see one is, if you use light microscopy, you can see the structure. When you look at under the fluorescence microscopy, based on the fluorescence, you can see the expression of your target. So now you can choose based on the structure, you wanted to isolate that particular cells, or if you use a fluorescence microscopy, wherever the fluorescence your target is expressed, you wanted to collect that. So that is, so our goal is whatever you are seeing under the either fluorescence microscopy or light microscopy, the interested cells you can able to isolate using this technique. So the transfer of larger laser energy to a thermolabile polymer with the formation of a polymer cell composite. That is, if you are using infrared or photovolatilization of the cells surrounding a selected area. That is, if you use ultraviolet or UV cutting system. So this is two principles how you are going to collect the cells. So our goal is removal of these cells, from the heterogeneous tissue section. That's our goal. So our goal is you are collecting a single cell from a heterogeneous mass of tissues. That's your goal. So what are all the sample types we can use? So here, when the source, the sample, specimen sources is surgical specimens. Either it is a frozen or paraffin tissue section. Cytology specimens like a touch probe, direct smears, thin probe, cytospin, cell block. So these are all the two different specimen sources. Then from that you can process your cells. The cytology specimen is you can thin smear on the slide or a thin probe. You can centrifuge them, the cells, and then you can drop them onto the slide and heat fix it, and then you can go for isolation. So then once you are transfer that onto the slides, and then you can stain, that is based on if you wanted to go with the H&E staining for based on the structure, or go for fluorescence if you express your target cells you could isolate. So that is the goal, and this is the targets, your specimens, and then once you are stain that one, and then use laser capture micro dissection to isolate the single cells from the heterogeneous mass of cells. So once you are isolating the cells, what are you going to do with that? That's most important thing here. Your goal is, one is look for the structure or target. So now you are talking about whatever cells, if you are interested to study further with biochemical or molecular level, you have to isolate that particular cells of your interest. So once you are done with that, what are you going to do with? We can do many things. That the application is studied, genomics or protein expression, RNA-seq or proteomic, and there are so many, target you can study. We will see that one by one. So specimen preparation. So here, the laser capture microdissection, there is no specific, preparation. Whatever you are preparing, samples for light microscopy or fluorescence microscopy, you can use that cells for laser capture microdissection. So all the process up to staining is same, either it's a light microscopy or fluorescence microscopy. After you place them on the stage, now your choice is whether you are going to just to see the structure or expression, then you have to cover them with the cover slip and seal them so that you can protect from dust or dry dust sections. But if your target is not only just to see the structures or expression of your target, but also you wanted to isolate your interested cell type based on structure or expression of your target. That means you should not, cover slip your tissue sections, because your goal is to isolate the cells. That means it should not be covered. So it should be as it is on the slide. Then you can, isolate the cells using the laser capture microdissection. So now the preservation is two different ways. One is chemical fixation, the other one is cryofixation. So if you fix your samples with the glutaraldehyde, that is, formalin fixed, that is a paraformaldehyde, that is used for, you can able to isolate the DNA from these sections. Whereas if you isolate RNA and protein, the best choice is a frozen tissue section. So the next is a stained or unstained, that is based on what your target and how. So that is, as mentioned earlier, that it is whether you stain with the hematoxylin eosin or immunohistochemical analysis, that is a fluorescence target. So during this process, we discussed about dehydration. So the dehydration is very, very important place in microscopy, especially when you are isolating cells from heterogeneous mass. Using this technique, the dehydration, if the water is present, that will reduce the quality of RNA. So the frozen sections, already we know that liquid nitrogen is fix your sample if you are using cryofixation. So once the OCT is a compound that is medium, the optimal cutting temperature. So that is at the room temperature, it is a semi-liquid, but when you bring that into minus 20, it become ice. So usually, like at the frozen sections, you can fix it in liquid nitrogen, and then you can put it on OCT medium and store it minus 80 for if you are not right away to isolate the cells. If you leave it for longer time outside, the RNA will be degraded. Then you can take a sections using cryo cut. Then again, after that, you can preserve that in minus 20. So the sections should be, 8 to 10 micron thickness. Again, after you take a sections, it will be put them back into minus 80 degrees centigrade. That will help you to preserve the RNA. So you cannot repeat, put on back and forth with the freezer and room temperature. That will also affect the nucleic acids quality. So the formalin fixed paraffin sections, that is another chemical fixation. That is also another, approach you can do. So if you are isolating your DNA from, the cells, the formalin fixed cells is also good. It's cross-linking for aldehydes, which affects nucleic acids. Thickness, when you take a section, so 5 to 10 micron thickness is, good to isolate the cells. After sectioning, we can store it room temperature, not like the, cryofixed cells. Then, once after section, when you take a paraffin sectioning, you can leave it in the room temperature until further, experiments. You could, not only you are taking use the tissues to isolate the cells, but also you can do cell suspensions. That's you can use smears prepared by centrifugation, or you can fix that with the ethanol, and then immediately you can isolate RNA from these cells. Also, you can store in the desiccator if you are using this in DNA isolation. So now, how we are going to isolate the cells. So this, figure which shows the schematic, steps how we are going to isolate cells from heterogeneous mass. As we know that after you process your cells and fixation, dehydration, infiltration, embedding, then sectioning, staining. So now your section is on top of the slide, right? You transfer onto the slide, then you process. Now you are going to observe under the laser capture micro dissection. Now you can see. So this is, imagine this is your slide. On top of that, you can see your section. Now, which is, when you see under the microscope, you can see the structure of the cells. So next is what we can do is we placed a thermolabile polymer of the caps and touch this bottom of the polymer onto the section. So now we can enable the infrared light or UV light. So that will pass, through this thermolabile polymer. So now this polymer is melt due to the transit, heat generated by the infrared or UV light. So this, polymer is melt. Immediately once it melt, it will attach to this area where we wanted to isolate. So when you look at this, this is, the area it melt, the polymer. Once you enable the polymer, it melt wherever it comes. So now this area is now melted, and which is attached to this section. When you look at this, when you take it out, you can see wherever, this cell type is attached to the thermolabile polymer, that is, generated heat by the infrared. So now you transfer this section onto the Eppendorf tube or any other container, and then you add a appropriate reagent to lyse the cells and do the subsequent analysis. Okay, so here when you look at this, this is how this infrared generate, and you can see. So here you can see properly melted polymer spots. So wherever this in the spot, this area cells will be picked up. So the protocol is typically the LCM, which utilizes an infrared laser integrated into a standard emitter microscope. A transparent cap is attached to a thermoplastic transparent membrane, which lies directly on the surface of a routinely prepared tissue section on a glass slide, as I mentioned earlier. So if you really wanted to take a, examine which part of the section, you wanted to do, we can activate that area using the laser when the desired cells underlie the target. You may ask one question that once it generates heat by infrared or UV, does not degrade the protein or nucleic acid, or the cell structure is damaged. The question for that, the answer is, this in turn activates the membrane with the subsequent binding and procurement of the cells of your interest. So during this process, the laser pulse is very brief and the membrane is activated at 90 degrees centigrade. Thus, the tissue experiences only a brief thermal transient. So this is the main, for the answer is, it generated 90 degrees centigrade so that it melt the polymer that stick onto the tissues, and then you can pick it up. During that time, that 90 degrees centigrade generated by the infrared is a very brief thermal transient, as the heat generated from the membrane is rapidly dissipated. So that is why you cannot, it does not degrade or affect any nucleic acids. So the LCM allows for visualization and the image capturing of tissue as it is microdissected, including images of the tissue before and after microdissection. So the advantage of this is we have a monitor that first you can photograph where you are going to isolate or which cell you are going to isolate. You take a photograph, and then after you isolate the cells, you can take another photograph. So now you have before and after you isolated the cells. So this data will help you in the latter stage that which area you are going to, which area you were picked up the cells, and what cell types you picked up, and how much you fixed up. So you have that evidence. So this is critical in maintaining an accurate record of each dissection and correlating histopathology with the subsequent molecular analysis, so that you know exactly when you have a final results, you can make sure that the isolated target is right or not. If there is something unexpected analysis you will get, then go back and check really that is the cells you are isolating is right, or different cells you isolated. Once the tissue has been properly processed, sectioned and stained, it is ready for microdissection. The laser beam size, how much area you are going to isolate, that is depends on the spot size, the laser beam size. So you can adjust the area of, from 7.5 micrometer to 30 micrometers to allow micro dissection of either group of cells or single cells, which is depending on the need of our interest. So once you isolated the cells and then you go for analysis. So how do you prepare for this? That's another important during this process. So already we discussed about this in the light microscopy and fluorescence microscopy. Here we need to take some extra steps. So for slide preparation is so important in this process, because you are not only just observe the structure, but also here you are going to isolate the cells for molecular analysis. That means you are going to isolate protein, DNA, or RNA. So that means you have to take an extra care. In preparation for LCM, the tissue of interest must be sectioned and placed onto a microscope slide, right? However, in order to allow for cell procurement, the sections must not be cover-sleeved, as I described earlier. At various stages of protocol development, the use of various slides type. So it includes, different slide preparation and the slide quality is important. So one is if you are taking sections using cryomicrotome, then the superfrost plus slide is ideal. So you can use the silane or silicon coated slides is also good. So once you isolate the cells, then subsequently you can isolate the DNA, RNA, protein from that. So the laser capture microdissection and subsequent molecular analysis can be carried out on slides stain using standard hematoxylin and eosin method or the fluorescence methods. So already we discussed about the staining, this. So, first you have to deparaffinize and then rehydrate the sample, and you stain with the hematoxylin, eosin, and then again dehydrate the sample and observation. Similarly, when you take a sections, you can look at the fluorescence microscopy and see, all the expression and process. So this is two different methods that you can utilize this, based on the structure or the fluorescence. So once, taking the sections, you can store that to room temperature if it is using paraffin sectioning. So do not deparaffinize until immediately prior to micro dissection. So once you take a sections, leave it room temperature, then wait for whenever you need it. If you take a sections, cryo section, you can store it in -80 degrees centigrade as we described. If the section was a frozen embedded, melt it gently for approximately 30 seconds after removal from the freezer. So this will create a rougher tissue surface and allow for better adhesion to the laser capture microdissection. So now, as I discussed, this is a deparaffinize, DNase, staining, and again rehydrate, and then air dry, and then let them go for laser capture microdissection technique, that is to isolate single cells or homogeneous cells, which is isolated from heterogeneous mass of tissue samples. Okay, so I will stop now here, then we will meet next class. Any questions, anything so far?